This is a moment we've been waiting for 1 year. Yes, it is true. At the last General Assembly of the ETPN, we made this old-fashioned survey on a piece of paper. What are the key priorities you want for the ETPN in the coming year? And more than, almost half of the people put regulatory as a key action for the ETPN, that things we don't do, we don't address, and are becoming a key priority. They want us to be a hub of information. They want us to play a role in this regulatory landscape. And we want to start by simple action. We will publish a web page with basic things, let's say regulatory for dummies in nanomedicine. We are creating a working group at the ETPN dedicated to regulatory issues. We will participate in the Metrino project about standardization, metrology for regulatory aspect of nanomedicine. And it was clearly written two things. We want a regulatory session in NME23. We want to have time to speak about these aspects. And the other is, well, please, regulatory, but not boring. So, yes. So we decided to have this very interactive session this afternoon, chaired by Professor Scott McNeill, notably from University of Basel. Then we have another talk from Dr. Where is Ka-Wai? From Ka-Wai Wan, yes. And it would be fantastic. And then we'll have a very interactive session with you and with some colleagues that are online. I hope they can hear me, notably Laurent Lévy, notably Arpan from Nanovation, and Sally Price from Evox. I'm looking forward very much, and I'm here to ask the dumb questions. Scott, the floor is yours.
Thanks very much. No pressure. I'm supposed to, in the next 25 minutes, teach you everything I know about regulatory science. So, gosh, I hope you can stay awake on this one. I think I'm going to hide behind here. That way I can do animations and stuff. Okay, well, first, thanks for the invitation. It's great to be here in Liverpool, interact with you guys, and see old friends and make new friends. So I bring you greetings from Basel. For those of you that don't know, Basel's in the corner of Switzerland, France, and Germany. University of Basel was created in 1460. No bragging rights there, but yeah, it's pretty old. And of course, we're home to beautiful scenery, and it's a hub of the pharmaceutical industry. We've got Roche there, Novartis, Lonza, Moderna. So it's a very nice place. Here's my acknowledgements and disclosures. I'm a member of many working groups. And I'm indebted to the NCL, the Nanotechnology Characterization Lab, where I was at for the previous 15 years, learning everything I know about regulatory science. My endowed chair is sponsored by CSL Vifor, and I also consult for Inomedica in Switzerland. Sorry, we have to post these. Okay, well, this group is no stranger to the capabilities of what nanomedicine can do. There's literally hundreds of thousands of papers on the capabilities of nanomedicine. So we can take insoluble molecules, hydrophobic, hydrophilic molecules, one with very, very high ρPs or partition coefficients. And we take those and we can formulate those in nanoparticles. So APIs, meaning small molecules that were once thought insoluble or unformulatable, we can now stick in a nanoparticle. So what this does is we can alter the pharmacokinetics, or PK. We can take that molecule and do a study on it in something like Cremophore, take that same molecule and put it in a nanoparticle, and change the AUC, change the PK, change the half-life. We can do all of the above. And if we're lucky, we can also modify the tox profile, where the small molecule may have a certain tox profile. We can decrease that by protecting the molecule in a nanoparticle. And what's really great is we can go the opposite way too. If we know a liability, a drug liability, we can say, hey, is there a nanomedicine solution to address that liability? Whether it's kidney toxicity, it's usually toxicity, if we want to increase the half-life and so forth. So we can take the API and then match it to the nanoparticle and go both ways. All right, so this is about regulatory sciences. And the first part of meeting the regulatory science and regulatory agencies is to make sure that we have a data package in hand. Certainly we can do a pre-IND with FDA or a pre-meeting with EMA, but that needs to be an in-depth conversation, and we owe it to them to have the data in hand. So what does that data look like? Well, the NCL has done actually a pretty good job of pointing you in the direction of some of the things that the data package that we'll need in order to have that pre-IND question, rather than just a conceptual discussion. So under PCC, physical chemical characterization, here's some of the things that most of us know. We're going to do size distribution, stability, and purity. This is, again, PCC. So this is just in PBS and water. This is not in biological media yet. But then we also do immunological characterization. What we're finding is many of the nanoparticles can activate complement. They can amplify an LPS response. Thank you, sir. And they can cause even cytokine storm. So we have a number of in vitro assays to look at that. And of course, we do PK tox. Even before we go into the very expensive GLP tox, we can do tox in most of our laboratories. Keep in mind that this is more than just 6 animals and they didn't die, so therefore it's non-toxic, okay? So that may work for the paper, but the discussion we're having here is how do we get into those clinical trials? And under biological characterization, I put this out here because we really have to nail the in vivo efficacy. And that's more than usually just more than a single experiment, cellular-based experiment. We want to absolutely be confident about our efficacy before we go to the FDA, and certainly before we go into clinical trials. I say this and I emphasize it because I've seen too many companies where there's some hand waving on this, and they'll say, oh, well, we'll get it when we're in phase 2. And they failed, and that, you know, multiple millions of dollars later. All right, regulatory considerations. We know where we want to go. We want to go to mountains in Switzerland, right? We know that destination. We know that we want to go into clinical trials. We know that we want to have a product, and we want to have some patient benefit, of course. So how do we get there? Well, actually, we have a map and compass already in place, because we're not the first as of today. There's multiple dozens of nanoformulations that have walked this path before you. So with a simple map and compass, we can pretty much find the point. Okay, so in the next few slides, I'm going to show you how to do that. You have a map and compass. I can't teach you how to not walk off a cliff. I can't teach you how to not freeze to death if it's snowing. I can't teach you how to avoid being eaten by bears, but at least we have a direction to head. All right, so let's say I have a nanoformulation. I have to choose which pathway I want to pursue. Either I'm doing both FDA and EMA, and this is all for an IV injection. The discussion, is it a device or is it a drug, is a worthwhile discussion. I'll just say that the regulatory agencies will determine that based on the primary mode of action. So if it's chemical, meaning if the drug hits a target, it's likely a drug. If it is implant, where the main mechanism of action is mechanical, they may view it as a device, but it's also in consultation with the drug centers, CDER in the case of FDA. All right, so which pathway do I take? What is a pathway? Well, the FDA and EMA have pathways in place, and there's quite a bit of literature on exactly this point. So there is no getting around if I have a novel API, meaning a small molecule, and I put it into a nanoparticle, I have to do a full phase 1, phase 2, and phase 3 trial and submit a full dossier. That's an Article 8.3 under EMA or a 505(b)(1) under FDA. With some luck, maybe I'll get through phase 2 and I'll get exceptional use and get it approved, but the standard way is phase 1, phase 2, phase 3 under a full dossier. But let's say I don't want to do that. I just want to reformulate doxorubicin. Well, actually liposomal doxorubicin. Well, then I can go the generic pathway, which is, as you see there, Article 10.1 or 505J. The cost for development of a full phase 1, phase 2, phase 3. To get into phase 2, we're usually looking at 30 to 40 million euros. And to do a full efficacy trial in phase 3 can be hundreds of millions of dollars. So there's quite a bit of money under full dossier. But in a generic, there are generics, at least in the US, that have passed without doing a single in vivo study. It's strictly a characterization, physical chemical characterization of the molecule, and they got through. That's not going to happen with a nanoparticle, but it can happen. It's far easier if you have a generic. But in a minute, I'm going to show you why not to do that. All right, there's also follow-ons. Now that the patents have expired on many of the nanoparticles, such as Abraxane and Doxil, we're now having follow-on versions of those. So let's say that I have an approved drug, and such as doxorubicin, the actual small molecule, and I want to stick it into my nanoparticle that's a polymer based. And with my polymer, I can show changes in PK or area under the curve, and I really want to push this forward. Well, I can do this under a 505(b)(2). It's an abbreviated drug application, and many times that just involves a bridging study where I can compare that to the approved drug. In some cases, you can do that with just a few hundred patients as opposed to the full phase 1, phase 2, and phase 3. So the cost for these, it depends on what that bridging study looks like, and also the imaging that's required. If it's a cancer drug, we have to show efficacy, and generally that includes things like MRI and CT scans that can be very costly. All right, so let's dig a little bit deeper into the deep end of the pool, take a dive. In order to do that, I have to show you some definitions. Definitions such as the active substance or active ingredient, depending on whether FDA or EMA, and the definition of excipients and inactive ingredients. And you're saying, McNeill, come on, we just had lunch, we're tired. Keep us awake here, you're giving us definitions. Well, hopefully you've had enough coffee, and just stay with me for 2 slides. Are you still awake? Are you still awake? So what you're seeing here on the left-hand side are the active substance or active ingredient definition. Below it is the definition of excipient. This is all online if you want it. And the difference between EU and US, I'll make it easier for you. So the active substance is intended to furnish pharmacological activity. Okay, for the US or for the EU, same thing, intended to exert pharmacological, immunological, or metabolic action. All right, it's pretty simple, right? Active substance, active ingredient. And then excipients is everything other than that. It's that simple. So we want something with a pharmacological therapeutic effect, and everything else, if it's just a sugar or a lipid in the formulation, is considered an excipient. So why do we care? Well, on the left, I'm showing you how a conventional small molecule is regulated. The characterization for a small molecule can be something as simple as LC-MS, a mass spec, to show, or an NMR to show purity of that small molecule. And so the biological activity of that entire formulation is based on the active substance. As long as I can very, very precisely characterize that small molecule, I know how it's going to behave biologically if I make it over and over again. That is absolutely not true for nanoparticles because they are complex drugs or non-biological complex drugs, NBCDs. So because they are complex, we're not going to make them exactly the same way each and every time. There's some boundaries and freedom in size, in the stoichiometry of the lipids, the loading, things like that. So characterization is not exhaustively possible. So then the biological activity is dependent on very complex parameters. Because we can have altered PK if we change the size too much or if we change the dissolution too much. And so this brings up the question, well, then what is the excipients and what is the active substance? And why do I care? So if I take a nanoparticle and run ADME-tox on it, that's pretty well established. We know most likely that it's going to be taken up by the organs of the RES, or more specifically the MPS. We've even seen data on that yesterday and day before. And it's feasible to have a half-life for a nanoparticle that can be 6 hours or even multiple days. If I take the composite nanoparticle, this is the result I get. But the current legislation, current regulatory requirements mandate that I evaluate that particle component-wise. So if I have a new polymer or a new lipid that has never seen human trials, we're required to evaluate that each and every component apart from and piecemeal apart from the composite nanoparticle. So that greatly changes the ADME-tox profile. ADME is absorption, distribution, metabolism, excretion. Because that polymer, separate from a composite nanoparticle that's 100 nanometers in size, may form a micelle by itself, a very small micelle. It may stay as a monomer, and it can be excreted through the kidney within a matter of minutes. So the tox profile can be completely different for the excipient compared to the composite particle. For example, if I have an area under the curve that is 6 hours or more compared to something that is only 10 minutes, I get dramatic differences, and I can have very different tox profiles as well. Okay, so what's the impact? Is that just a nuanced difference in regulatory terms? Why do I care? Well, there is a definite commercial impact. If I have a small company and I've got a burn rate that's going, and all of a sudden I find out that I have a 2-year genotoxicity study before I can go into humans. What did my investors just do? They're going to be on the phone with me. Hey, you promised us in 12 months you're going to be in clinical trials. So they're expensive and they cause delays in the programmatics. But as I mentioned, they can also generate misleading results. If I get altered PK, PD, tox profiles, and so forth. So how does this impact the regulatory dossier? By the dossier, I mean the paperwork that is used to file to the regulatory agencies. Well, there's now a common technical document, CTD, that is shared between the member states, as well as I think the FDA 2 years ago also adopted this. It has 5 main modules. The ones that we most care about are the CMC and farm tox, chemistry, manufacturing controls, and the farm tox profiles. Those happen in module 3 and 4. Why am I telling you this? Well, stick with me. It's because the developer, meaning I as the investigator, initiate the classification of whether I want to call something a excipient or the active substance. It's me as the developer that makes that classification. The regulatory agencies can either then accept it or reject it or talk you down, but the regulatory agencies do not initiate that classification. It's on us as investigators. This is a very important point, and I'll show you a case study on this. Okay, here's some examples of the LNPs that we've seen many times in the last few days. The case study, I'll compare Onpattro, and the two mRNA vaccines. Keep in mind that Onpattro is an IV administration, and the vaccines are obviously IM administration. Well, they're very, very similar as far as composition and lipids. If you take a look at the chemical structure, you'd say, hey, those are the same. There's one branched in one of them, but otherwise they look very similar. But look at the differences in the classification of those products, despite the very, very similar lipids. For EMA, the lipids, all the lipids were considered as excipients. But for the FDA, the lipids for the Spikevax was part of the drug substance. How did they do that? Well, they did what I just showed you. They initiated that classification. It was Moderna had a very strategic insight on how to get this done. So they listed it in the US as part of the drug substance. FDA bought off on it, and boom, they were off and running. Whereas Pfizer and Onpattro did it the traditional way, thinking, hey, we know what we're doing. We're just going to pursue it like we've always done it before. We got a new excipient, blah, blah, blah. Let's go that pathway. So it was very strategic. And I point this out because the compounds are exceptionally similar, and it was all initiated by the developer, meaning Moderna. All right, so what does this mean as far as the rest of us? Is it just a nuanced regulatory terminology? Well, no, it has applications for us because it sets a precedent, a baseline. We can always go back to that example, and even in a legal case, say, well, wait a minute, you did it like this for Moderna, why don't you do it for us? Here's the criteria that we can use. And then what's next? Well, we'd like to somehow codify this so that there's no ambiguity when we approach the regulatory agencies. We don't want to approach them and find out that we've got a very extensive GLP tox study that we're required to do. We can get that decision made even at the pre-IND level. And I put up here regulatory sciences, which is half of my field. We have these types of discussions because we can meet with regulatory agencies, we meet with investigators, we can meet with the producers, CDMOs, and come up with a consensus, a common ground where we can discuss these types of problems. Here's the paper if you're interested in looking at it. It was just published, I think, 2 months ago. It's available. All right, so now you know everything I do as far as regulatory sciences and regulatory considerations. But wait, there's more. The other half of the equation that I'm trying to balance so that I can get my product commercialized is my reimbursement. It's the role of the payer. So what role do the HTA's health technology assessment bodies play in clinical trials? I didn't even know what this stood for until about 2 months ago, and I'm learning how very, very important it is. So these are the guys that once I have a product, they reimburse me for whatever I made. It's the insurance. Generally in Europe, it's state insurance. Okay, so let's go run through a notional example. Let's say that I develop a nanomedicine that has 5 times the AUC, area under the curve, the integration, in a phase 1 trial compared to the standard of care. Man, I'm on cloud 9, right? I'm making the cover of Nature Nanotechnology. I'm patting myself on the back. I'm paving a way for future investments and clinical trials. But the real question is, can I get a return on investment to support the very expensive development of this product? Because it works very well, but it's also 100 times more expensive to produce than a small molecule as a conservative estimate. I mean, we even saw that with AstraZeneca, where Marianne presented yesterday. Okay, so McNeill, you just told us that the end goal is no longer commercialization, but it's reimbursement. It's not patient benefit. You will hear that from the payers. It's the cold hard facts. Is it worth paying you such and such amount? But even now we have the ability to crunch those numbers. This was very surprising to me. We can look forward based on the current prices for the standard of care and look up a table. And if my product does not offer an advantage compared to that standard of care, the most I can get is 75% of the current standard of care. That's generally throughout Europe. All right, so the standard of care in Europe for ovarian cancer is doxorubicin. It's not Doxil. It's not liposomal doxorubicin. It's Doxil, right? That's free doxorubicin. The price of a vial of doxorubicin is 14 francs in Switzerland. And I'm only allowed, if I'm a physician, I'm only allowed to prescribe Doxil in a case of adverse side effects. My go-to drug costs 14 francs. My liposomal doxorubicin is 240 francs. Okay, so which one do I compare to? If I have a novel formulation, I actually compare to free doxorubicin at 14 francs per vial. That's what we're up against. Okay, so this is a problem, right? Because if I'm creating something very expensive, I want to be reimbursed and make sure that I have something that the investors can invest in. Well, the question as we approach the HTA is, how does your product decrease the financial burden on my public health system that I represent as BAG in the case of Switzerland? And we have to be able to speak it in these terms, because if my response to this question, when we sit down with them, is, well, wait a minute, I have 5 times the AUC. If that's my answer, and I'm going to be defensive, I got the cover of Nature Nanotechnology. How dare you ask what the advantage is? Well, they think of it in terms of the reimbursement is proportional to the patient benefit. So if that patient benefit is minimal, don't expect a huge multifold reimbursement compared to the standard of care. Well, my second answer can be, well, I don't really care because my business model is I'm going to sell it as soon as I exit phase 2. How many of you have heard this before? This is the standard business model, and it decreases risk for the pharmaceutical industry. But let me tell you, the pharmaceutical industry has a team of at least 10 people working these numbers to see if they'll get their return on investment if they invest in you or license your product. So option 3, coming back to regulatory considerations, is I have to make sure that my phase 1, phase 2, well, mainly phase 2 and phase 3 trials justify what my claims are. It expands and exploits my innovation. So if I have 5 times the AOC, I have to put that in terms that the payer can understand. And that can be as simple as when I do my phase 3, if I am saying that it's more efficacious, then I have to show better efficacy compared to the standard of care in my phase 3 trial. it becomes very expensive, but I can't just notionally say, well, I've got better PK, give me more money. It just doesn't work. And we do this in collaboration with the regulatory agencies. We're not required to meet with the payer without clinical data. I mean, these guys are talking with one another, so it's a at least in Switzerland, it's a decent conversation. All right, so the peak that I showed you before is a nice peak that many people have hiked, but really, if we want to make a difference in nanomedicine and we want the preferred return on investment, we have to go places where no one has gone before, such as orphan diseases and exceptional needs. Without that, the money is not looking good. And this is somebody who's been in the field for 20 years telling you this. We have amazing capabilities. You have the ability to offer a product for diseases that there's no cure. I'm working with lysosomal storage diseases, and Pompe kids, if they get ADA, anti-drug antibody, that's it. They have no cure. But nanomedicine has the ability to encapsulate the enzyme and offer hope to these orphan diseases. Okay, so there's the destination, even in phase 1, even in preclinical, know where you're going, and plan your route. If your approach is, if I build it, they will come, they're not going to come. All right, you have to show them in terms of better efficacy, better decreased toxicity, so that the pharmaceutical companies will invest in you, and the payers will reimburse you to that level. Let me acknowledge Eva Hemrick, who is really doing the heavy lifting on most of our regulatory sciences. Here's our contact info. And let me also make a plug for 2 conferences that are coming up in Switzerland. First is Nano GVA in October, and immediately following is Clinum there in Basel. So with that, thanks for your time, and I'm probably, well, I got a minute or 2 for a question. Okay, thank you.
Indeed, we have the panel to go into a dense discussion, but see if you have some questions for me, for Scott. Yes, indeed.
Great talk, Scott, as always. Just 2 questions, if that's okay. So first one, I forget the EMA nomenclature, so apologies for that. But can you summarize for us, just at very high level, the difference between the 505B2 and the 505J?
A J is generic, J for generic. That's how I memorize it. It is the exact same formulation as one that's already in the market. So if I want a 505J on Doxil, liposomal doxorubicin, I have to make it exactly PCC, everything compared to Doxil. A 505B2 is where I am offering some new capability against the same drug, meaning I'm still got doxorubicin, but I'm putting it into a new cargo, new vessel that has different release profiles, decreased toxicity. So it's when I tweak it, I go the B2 route.
So if it were the same carrier, different process for manufacturer?
Say that again.
A same carrier system, different process for manufacturing? Where
You're talking about for the J?
Well, would it sit in 505B2 or would it sit in J?
Yeah, so for the 505B2, if I understand you correctly, I can do anything I want to. I can, as long as I got that small molecule, I can put it into anything I want to, as long as I don't covalently, as long as I don't change a covalent bond. Am I answering your question? Yeah.
A second question, really quickly. Why didn't Moderna go the same way with EMA as they went with it?
That's a great question. What they tried to. They tried to go the same way with EMA, and they tried to go the same way with Japan and Asia and others. EMA said, no, not so fast. Yeah, that's exactly what happened. And you can read it. I mean, it's open source information. In fact, on our paper, I think we cite the references for that. Yeah, EMA caught them. Please.
Thanks, Scott. I could listen to that all day. Brilliant. Got loads of questions, but I'll pick one. What is the benefit really of going ahead with it with a lipid or a novel excipient classified as part of the API? Is it that you get to, reduce the amount of tox studies, you're studying the thing as a whole? But then it raises the question of what if you're trying to go for like a platform type system? Then what do you do?
Yeah. So the first part of your question is, why do I want to put my nanoparticle that I've developed where I have IP on it into clinical trials in the first place? That goes into my earlier, one of my first slides, is there's some capability that I have with that novel polymer that maybe it incorporates a hydrophobic molecule far better than a liposome does. And so I can offer that as a capability. So it's worth pursuing my novel lipid, my novel chemical entity, NCE, because of that capability. If there's no capability, then it's not worth pursuing.
No, I mean, rather than having it as the excipient where you said you have to test the individual components, but you could alternatively try and classify the whole particle with all the components as the API.
Exactly. Yes, that's where we're headed. That's where McNeill is pushing hard in exactly that direction. The current policies are a little bit ambiguous in that, because the determination of what you just said, active substance or excipient, is made by a very small committee within the regulatory agencies. You can influence that and try and make the scientific case, but ultimately it's them. Yeah, I can classify it in my dossier, but they get the final say. Okay.
Get your brains on this afterwards, I think.
Yeah. Now, the second part of your question.
Well, it's very related, because if you're going for a platform system, so if you test it as a combined API, your mRNA plus a particular carrier might be a lipid, but I'm thinking something else, perhaps like a cell penetrating peptide or something. If you classify that whole thing as your API, your active ingredient, but then you're trying to take a platform approach, so you've got a different mRNA plus the same carrier, then what do you have to do? You're not saving anything, whereas you might have done if you did it as a separate component.
Yeah, that's a terrific question. And that's exactly where the field is right now, as far as the debates and conversations. Can I make my vaccine modular? So I'm just swapping out the mRNA, it's a different sequence, and use the drug master file on all the lipids. I think that they're pushing that. I think it's going to happen, but it's not going to be simple, because even though mRNA is mRNA, it's a different sequence, right? Different target. And so they're going to want to make sure that there will be some sort of in vivo studies to make sure there's not toxicity associated with that. That's active debates right now.
Thank you.
Behind you.
Can I just add to that one?
You're the speaker. So the speaker asks a question.
I'm not asking a question. Can I just answer the lady's question? Hi. It's burning, okay, because I wanted to say something before it gets too late. With regards to Moderna's proposal as the active substance, I must say the regulators are absolutely disagreeing on that from the EMA perspective, MHRA, Health Canada, globally, perhaps except FDA at the time, but I think they're also changing, because it would actually cause upheaval of the underpinning fundamental regulations of what is a drug product and what is a drug substance, I'm afraid. Think about, some of you may be scientists, pharmacists here, formulation, drug delivery systems, liposomes, lipid nanoparticles, all of these are considered as the formulations. The drug product is a drug carrier, helping the drug to deliver to the target site. If you consider the drug delivery system as an active, then we need to change all the liposomal doxorubicin, all the products, albumin, as the excipients, changing it to an active. And understanding the active substance definition, as Scott kindly showed on the slide, its pharmacological effect. What is the mechanism of actions? You could argue some of the adjuvants effect being used in the lipid nanoparticles may have some immunological effect. But in vaccines, even adjuvants, which are adjuvants giving you immunogenicity, are not classified as the active. They are still classified as the excipients. And those have been historically being treated as adjuvants. So we can't causing upheaval of the underpinning science, the regulatory science in this aspect. And fundamentally, it's not just about causing inconvenience to the regulators, it's actually the science behind it. Because regulations for the active is different to the excipients. There are GMP involved as well. We have very rigorous QP declaration where you have to look at the manufacturing of an active, and we have GMP for the drug products. And these are all scientifically based. We would not want the product to get near the patients if they have not been inspected. You would agree with that. But if you actually classify them as the active, the manufacturing process depends on the starting materials. Anything before the starting materials are, strictly speaking, not regulated to the same extent as anything classified as a starting material. And that is crucial when we consider how we regulate the drug product in its whole entirety. I hope that clarifies your confusion between what is a drug substance and what is a drug product, because that is a heated debate.
Yes.
And I can tell you how many times I've argued with Moderna. They hate me, but my job is not to be loved, as I said. So there you go. So it's always the shortest person in the room that will give the annoying comments. But yes, it's a heated debate, but I really need to say that it is not an active. And so this would actually cause a lot of problems in terms of regulations, because we don't want it to be unsafe for the patients at the end.
Well,
That's the ultimate.
Ka-Wai is from MHRA, so she speaks with authority. But I do look forward to active debate on that exact topic.
I was hoping interactive session. It starts between the speakers. That's fantastic.
So I'll turn it over.
No, please go ahead. Yeah, that's right now.
So let me introduce Ka-Wai Wan, Dr. Ka-Wai Wan, formally. She is at MHRA, which is the policymakers, the decision makers, I should say. She's a senior pharmaceutical assessor, so she is the one that is reviewing the actual dossiers.
Okay. Distance. I was going to Zoom. Yeah.
Do have time, maybe just for one question. We're going to have a panel session afterwards that includes both of us as well as industry members. Quick question. Yes.
Yeah, great, thanks. So you spoke a bit about PEG, and I think it's an interesting situation at the moment where the safety profile of PEG is potentially changing. And I don't know how regulatory bodies are looking at that. Is there, are they looking, or are you looking back to look at previous PEG formulations and say, actually, we might need more safety profiling going forwards, or are the standards of what's acceptable going to change? Or yeah, my question is just about that, really.
Thank you for the question. PEG was actually another heated debate. There are many products classified in the medicinal products, not just in nanomedicines, actually contain PEG. whether it's in a cream and in an inhalational device, as well as these nano medicines. So yes, we are looking into that, but we are not in the process of saying any product containing PEG, you're not allowed to be used. We will still look at the in vitro tox studies. We'll still look at the immunogenicity, as you would with any vaccine products in particular, or any other products. You will still need to look at the non-clinical data and the clinical data. And for the clinical assessment team, they would always look at if there were any unexplained rashes, allergic reactions, and where are they being attributed to. So it could be PEG, it could be something else, but these are what we call signals. So if there are many signals which would actually warrant further investigation, then we would ask the company to justify and to explain what you're seeing in the data, and then further studies to be performed. So that will be on a case-by-case basis.
Okay, great, thanks.
Okay, I'm going to turn it over to Alex, who is going to be the chair of the.
Oh, the chair.
Session.
Yeah, I'm here to ask the silly question, but maybe before, they've been very patiently waiting for us. Are you still online and alive? Laurent, for instance, can you hear us?
Yes, I can.
Hi, Laurent. Hi, Arpan. Thank you so much. You are not alone. I mean, a lot of people are still in front of you in the room, and also Sally Price from Evox is just here to join us. Sally, please. You have the best one. Laurent, Arpan, simple question. Do you have any comments on the two first talks? Any thoughts? And yeah, please, let's start like this. Any reaction to what you just heard and the debate we're starting to have? Go ahead.
I can say a comment. I think it was really fascinating to hear that perspective. Just to say, you know, the perspective that I come from. So at Nanovation, we're very early stage research. So, you know, very much discovery focused in how we design lipid nanoparticles, and mRNA is what we're doing. And, you know, thinking about it from a regulatory perspective, it is a different way of thinking of things. And I think there's those talks really helped think about that. And I think the way we think about regulation, I mean, what I would say is, for us in our early stage, there's a balance. There's a balance between how much we really like focus on the minutiae of, you know, the critical quality attributes of everything we're doing, versus like going ahead and testing new things. And getting that balance right is something that we have to do. And like, to give you an example, I think regulation can be really enabling for what we do. And I'll give you one example where the insights from the regulatory space has really helped our research. And there was a very significant paper, I think, published by Moderna in 2021, which talked about how lipid impurities react with mRNA to form adducts, and that can degrade the RNA and have a big impact in the biological performance of the lipid nanoparticles and mRNA. And from that's something that we hadn't considered hugely before. We now implement stringent testing procedures on our lipids and our RNA to show it's not degraded. So I would say there's a lot to learn from both areas, and it's something that I think we need to be considering all the time.
Yeah, so can you hear me when I'm like that? Yes, you can. So a direct impact of the design of your product and solution already, yeah? In advance, let's say now. Laurent, maybe a first comment before we move on.
Well, I would say all this sounds familiar. I mean, we've been through many interaction in regulatory matters, and it's always fascinating. There's one message I would like to convey, but maybe that has been already done, which is every single product should be considered as a different product. And there are so many variations when we build nanotechnology-based product that it's very hard to rely on what has been done in the past. So when we do new product, we always look at what are the boundaries or what are the frame from a regulatory perspective, which is very helpful, but also as on step number 2, always looking at the specificity of the product and what this product will require from a scientific and medical perspective to be safe and used into patient. And so far, I did not have any equivalent in all the product we've been developing or seen. There's always something different that requires some tweak, some little different approaches, some different way of characterizing the product, some different way of looking at how we should establish preclinical efficacy and safety. And so I think adaptability is what we need to do when we look at regulatory matters in nanotechnology. And also, the more interaction we have with agencies in general, the better it is to understand and to most of the time co-build a pathway for a new type of product that prevents surprises on both sides and always help. At least that has been our experience talking with agencies, even before making the first chemistry around the particle, was very helpful for us in order to design the pathway.
Just a quick comment on what you just said yesterday at the next big thing in nanomedicine session. You've mentioned that in order to optimize your chance to be successful, you need to some kind of, let's say, avoid having one single solution for one single narrow application, but maybe more to think as a platform, you know, like a platform, a generic platform of technology for a generic platform of applications. But you've just said that on the regulatory point of view, every time it's different. So as a platform, do you learn something which is useful every time for the next one?
Oh, yes. It's not because they are not exactly the same that you can't learn from something else. For me, there's a lot you learn when you move from one particle to the other from a manufacturing perspective, which is a big part of the question or the quality attributes. So yes, you transfer a lot of know-how and learning.
And learning how to answer the question, maybe.
Yes.
Sally, maybe a comment on Evox point of view. You have the mic, I guess.
Yeah. Thank you. Yeah, so just thinking about the talks and the complexity, and particularly with EVs, because they're obviously made from cells, we have the biological complexity as well. Just a reflection from me that we as a company have gone to the regulatory agencies early, and that has been really, really valuable for us. And I think, you know, now for our future projects, it's actually thinking about the questions that you want to ask them as well. So, you know, you need to think about what is it you want to have advice on. And I think some of the things for us has been around, you know, sort of that technical detail, the methods. A personal one for me around some of the biodistribution work wasn't so much about the biodistribution, it was about the methods that we were using to do that, given that the different methods for EVs measure slightly different things. It's that level of detail that maybe I hadn't thought about before. And getting that early interaction, and it has been different things, even though we have a platform technology, it has been different things on different projects. So really agreeing with every case is slightly different, but that early interaction has been very valuable for us as a company.
Sally, can I ask a question? When you define early, in practical terms, when is early? I mean, can you give some examples, some practical examples without, of course, any
I think it's a really
information?
good question, because you don't want to go when you haven't got any data. And I think, so obviously you want to have done some work, generated some data, and from us, particularly on the CMC side, I think it was important to get some of those early questions done. But in some ways, if you go too far down the line, so particularly for us in terms of your cell line selection, your patient donor of that cell line, you can have problems if you're not careful that mainly go right back to the beginning. So it's a really difficult question to answer. But I think the other thing I'd say is, you know, speak to other people who've developed similar products or any products before. Because reading through some of our very, very early stuff before kind of more experienced people join the company, some of it you go, why did you go and ask that? It's really obvious. So, you know, use the people around you even before you go and speak to regulators, so you generate the best data you can for the stage that you're at.
And
Yeah, no, maybe because we wanted to go back to, let's say, some kind of basic questions that we all may ask, right? I guess we have a lot of academic people also. If I were a technology developer after such a session, I feel the expertise. I feel it's really complex, but I mean, we have guidance, but I would feel a bit like overwhelmed by the level of complexity, the level of experience, the quantity of parameters, the quantity of method to, I mean, deliver the parameter, etc., etc. So my question, I have two questions. The first one is, are you surprised that when we do a survey among our ETPN members, they are super afraid about the regulatory burden? That's the first question. Let's start by the basics. Shall they be afraid, or do you think it's just about getting in contact with you? Please.
The simple answer is, if you're already in nanomedicine, you have a personality that is tenacity, yeah? It's not for the weak at heart. So that also applies as you further develop it. You're going to get hit, but also it's very rewarding. The interactions are collegial with regulators, with our peers. Hopefully you've even seen that here in this meeting. So I would say absolute, my position is absolute encouragement. Go. You guys have got the cure for some things, for a patient that has no other choice, no other options. Run with it.
Thank you. Any comment, maybe Ka-Wai? People are concerned, I can tell you.
No, echoing what Scott said, for the MHRA or for us, basically, we want the patients to have the best treatment or therapy. So as long as you come and speak to us, we'll be able to help you rather than creating a barrier as such. I don't want that.
A quick question. Do you know, and if yes, how many of you have tried to contact directly the email? They have an office for academia and an office for SME. Who has contacted the office of the email for academia? Don't be shy. You have, come on. And you sit on the first row. Indeed.
I seek advice. It was for a topical product, actually. And I had
What, sorry?
It was for a topical product. And I actually had the same question, whether it would actually be an excipient. It would be classed as an excipient or as an API. And it did help me at the time. So a bit in the middle, I would probably say, from that interaction.
And on the SME point of view, we have lots of SMEs also here. Have you already tried, before having any data to show, to contact them to make the right choice from the beginning? So to let you know, you can contact the EMA. I strongly advise you to have a look at the replay of a very nice session from the EIC and EMA joint event that was made at the end of January 2023. You just Google EMA EIC, and you have precious information about the tools that are here to make the right choice. Okay. Hello, please. Indeed. Can you please wait for the mic, because otherwise they don't hear you. I told you like 5 times, Pablo, come on.
So FDA, EMA, they all have put in place these protocols whereby you can contact them, right? So the EMA as the Innovation Task Force is the right office to contact for our stuff. But the question that I have is, do you contact them as a scientist, or do you contact them as a scientist that is sitting, or as a regulatory scientist that is sitting next to you? Because I'm not sure how we cannot just drop them an email and say, look, I have this nanoparticle that is doing X and Y. I think that you had to put together some formal document. And I'm not yet at the stage of generating these formal documents, but I was actually wondering if that formal document can be written by a scientist, or you will still need a regulatory scientist sitting next to you, which means that you either have a friend or you have an SME.
So I personally contact them as a conference organizer, and they do not reply. But that's a separate topic. Yeah, but maybe Ka-Wai or Scott, you have a comment on that.
We look at the innovation inquiries, and we would like to see the science behind it, because it will be us looking at your queries. For example, if you're going to ask a question, is this going to be classified as a medicinal product, or is this going to be classified as a medical device? If you don't provide any information, I can't just Google and expect to give me a list of information that will help me to decide. So the more information you can provide to us, the more elaborative answer that we may be able to provide to you and give you some considerations to help you. That's the answer.
Next question that I have, very basic one. It's super complex to have the perfect data matching the requirements and to go until clinical development. Okay, but what would be like your top 3 advice to avoid the basic mistakes from scratch in the development of the project, somehow to be, let's say, regulatory compliant by design, not just safe by design? What would be your top advice? I mean, from you guys and also maybe from Arpan and Laurent. Who wants to start?
I think Laurent should be able to.
Yeah.
be first
Laurent, do you want to
answer that
start, Laurent?
question.
Scott gives you the ball.
Yeah, why not? Thank you, Scott. And by the way, hi, nice to see you. Well, I just want to react to your first question, maybe about being overwhelmed by our regulatory. I think most of us, if not all, are scientists. And when we started studying science, you just going to the university and you see this big book, Cell, you say, oh, damn it, I'm going to have to learn all this, how I'm going to do it. Well, regulatory is the same. It's like a new thing you need to learn. And you just need to go for it and to learn and to read and to talk with experts. And just like any other science, you will learn how this works and potentially will be part of creating a new path with regulatory agencies because you'll develop a new product. In that regard, the thing I will do now all the time, developing new product, is first really looking at CMC. When starting, and what are the quality attributes of the product that may influence the benefit-risk ratio for the patient. For me, this is so tricky and so complex going forward in the development and may kill a product that I always look at this at the early stage of development. That's 1. 2, I will always challenge not the guideline itself, but always look at, is it the right question for the product? For example, some products do not have pharmacokinetics because you inject that locally, they stay here, they don't have any metabolic pathways. So you need to re-challenge the notion of PK/PD for such kind of product. But so what could be the equivalent of PK/PD for such a product? What do you try to achieve? What do you try to show? So that will be a step 2. And step 3, for me, it's always talk with experts. There's plenty of people that have been working in nano in other fields and that went through that. So getting a number of experts, getting a number of discussion with agencies for me is a very big help. And it may cost a little, but a mistake in designing a trial, a mistake in designing a pathway could cost a lot more. And let's not forget that at the end of the day, what we develop will be at some point in contact with a human being. So that's a big thing and a big responsibility. So avoiding mistakes by design, I think it's a good pathway forward.
You want to react? Otherwise, I have a question. No? Arpan, Sally? No. How open to redesigning what PK and PD are regulators? I mean, how are you open to understanding differently something that should be so simple, so generic?
We are always open to innovation.
You can always send email. No, seriously.
Seriously. We always look at the evidence.
Please use this one, it's better. No problem.
Joking aside, we always look at the evidence. So whatever you present to us, we'll look at them and we'll make sure they meet the required standards. And that's just our job. So as long as you know that your data is robust and you can justify why you did the studies the way you did, and then there are clinical benefits at the end, then you'll be safe. But you only on very loose water, if you don't have a good, robust set of data, and you're trying to push the boundaries and say, this is safe, but in actual fact, you know, there are patients dying in your clinical trials, then definitely that's not going to work. So yes, we do look at the data, and we will make flexibilities based on the data presented in front of us.
About pushing the boundaries, there's another, let's say hope or fear, you choose. In this community, we often hear people that, I mean, Europe can show up with a brand new regulation only for nanomedicine, you know, because it tries to fit with a simple scheme, but it's too complex. So we need to reinvent totally the regulation. So impossible question. First, do you see it coming? Second question, do you think we should adapt the global framework? Maybe, Scott, would you like to comment on that? Would you dream about nanomedicine specific regulation that would fit with the complexity of the functional design of this object, which is so complex?
I don't see a guidance document other than what has already been published as far as regulation. So there is a guidance document on nanomaterials that's more or less an information paper that was published by the FDA that goes through many of the things that we've talked about. In addition, there are product-specific guidances, such as the liposomal doxorubicin, some of the iron carbohydrates. So I see many of those coming out and being revised. But the mantra that I've heard now for 20 years is regulators do not regulate technology, they regulate products. And I don't see that changing in the near term.
Are there questions from the audience? You are a bit silent.
Focus.
Oh, as you wish, I mean.
Okay, me first. Yeah, I was wondering a little bit about excipients. And when you bring in new excipients, will they require in a way their own dossier, or how do you evaluate those?
Actually, that was my question as well.
Excipients. Any materials that have not been tested in human will be considered as novel excipients. It would still be considered as novel excipients, even if it was used, but via a different route of administration. So that's something that perhaps some people may be surprised. So let's say the excipient has already been approved as an oral delivery, and you want to use it as an inhalational formulation. It will still be considered as novel excipient by its own administration route. So in that package, you will still need to perform the studies relevant to the route of administration, as in this case, inhalational. And so any novel excipient would require extensive in vitro testing. So the animal studies, tox, genotox example, just those as an example. Also, sometimes you see in vitro studies to try and correlate to the animal studies to prove that they are okay. And so we look at the full package. Genotoxicity, mutagenic, and sometimes whether or not the excipient will be used passing the teratogenic, so passing the placenta as well. So we also need studies on these, or data, to prove that they're going to be safe for the intended use. So that's important.
What are the requirements in terms of the manufacture of the excipient? So this is how the excipient will behave biologically, but do you impose, when it's a novel excipient, any package of information in terms of its manufacturing quality?
The product is authorized to the marketing authorization holder. So legally, it's the marketing authorization holder that is responsible for the product, not the excipient manufacturer. So you have the ultimate responsibility to ensure that the quality of the excipient meets the required standard. And you would have the specification of your excipients to be included in your dossier. And so you need to understand the manufacturing processes of the excipients, and you need to submit that in your dossier. In some territories, you would have the excipient like an ASMF, active substance manufacturing file. So we haven't currently in the EU setting got an excipient master file, but I sincerely hope one day before I retire, we'll see that somewhere, because I would love to have those master files so that we can translate into different products, and that would be acting like an ASMF. So you do need to understand the manufacturing processes, the controls, etc., and the shelf life, etc., and embed that into your dossier. Does that help? Yeah.
Just for the sake of it and then complicate it a little bit more. So what if you perform in situ polymerization when you make your particle? So then you're basically forming your end excipient in the process when you're making the particle. How do you characterize that, like you say?
That would be considered as part of the manufacturing process of the drug product, because you're actually making it with the drug products. So yes, the materials are being made, but the manufacturing process of the drug product is in its entirety. So you need to treat that as a drug product altogether. The actual raw materials is what you're considering, needs to meet required specifications. And you propose that with justifications and testing.
I need to do my run.
Thank you. Thank you very much. Yeah, sorry, Alex Kramer from Avanti Polar Lipids, just commenting on some of these statements. I think it's worth saying it's never too early to speak to your excipient manufacturer as well in the process. We will need to put a GMP process in place for you, depending on where you intend to register. We will need to potentially create an ASMF for you or support you in that process. So I think sometimes people find that a little bit intimidating, going and speaking to an excipient manufacturer, but I think people are pretty open in that industry nowadays and will be very willing to provide with advice on what's necessary and what the timelines are to take a novel excipient through that process from a manufacturing perspective.
Just want to add to that as well. Hearing that there's a bit of a barrier, it seems, from academia or the innovators to go in to ask for help. So not as a sales pitch by any sense, but there's folk like our company who, our directors and ourselves have got years of experience with big pharma, done loads and loads of regulatory interactions through to commercial. They know kind of what questions to ask, what questions not to ask, what to say, what not to say, and we can help to sort of put together the dossier and help to phrase the questions and things like that. Some of them we might know the answers to, as Sally alluded to, but if we don't, we can absolutely help to engage with the regulatory authorities. So what I'd suggest is make some space in your sort of grant applications for some funding to help to get that support. And not just my company, but there's loads out there that we can, help us out there, approach the catapults, things like that. We can do this together.
Most of the European funding asks for regulatory aspects, but they do not provide yet a clear pathway to do it. And maybe, we used to have a fantastic tool that was the UNCL to at least do help on the characterization with a cascade of, let's say, well standardized, but we don't have that anymore. So we try, but in the mic, please, Fanny, come on, not you, please.
Well, Angel was just presenting yesterday Safe and MedTech, the Open Innovation Testbed, one of the Open Innovation Testbed for the medical devices. I'm not sure if he's today in the room, but in that, for example, service, they plan also to have some regulatory support, and we may have also additional tool and think about additional tool to do that. And as Claire was saying, there are also companies that can help with that. But do you think that the ETPN should do something for?
We're fine. We're starting. This is, paving the way for a new action by the ETPN. But most generally,
I mean, I have the impression that now in European funding, you have to be translational. You have to be translational. And sometimes it's a bit delusional because you won't be, you won't become a clinical tested product. And I think it's a pity because maybe we should focus on the one that has really a chance, but to help them concretely with European-wide initiatives. Well, we will do what we can to help on that. We are close to the end of this panel. I would like to give, I mean, for maybe a final advice or a final question, the mic to everyone, maybe starting by Sally. I don't know. Last point from Evox, like for instance, if you would have changed anything in the process, knowing what it will be, what would be the pitfall, the mistake you would avoid if you try the second time?
I think someone's already said it before, that it's never too early to think about it. And I think going back to Scott's talk in terms of the payers as well, just be, you can go through everything, but if it's never going to be a product that you're going to be able to differentiate, then it's kind of all for nothing. So think about that at the very start, because there's normally lots of ways you can pivot. So think about that and think about all the things you've heard today. It's never too early.
So are you cheaper than doxorubicin? Okay, Arpan, I don't know. Do you have a last comment? As you're still developing everything, so.
Yeah, so I mean, I would say I'm not clever enough to give advice, but I would say that one thing that's helped me is to go in early and look at the EMA and FDA documentation for the products that's closest to what you're researching and what you're working on. And I think that can give a lot of insights into kind of what that product and that what the regulators may be looking for a little bit later down the line.
So as Laurent, you go back to school when you want to develop these products, right? We need to learn a lot, right?
Always.
Laurent, words maybe?
Well, I mean, that's a lot of work. I don't see challenges, but a lot of work. But that's a good work, because at the end of the day, we can help patients with that.
Good final words. Maybe Ka-Wai, do you have a final comment to close the session?
If you have any questions, please just come to see us and we'll be happy to help.
Fantastic. Scott, would you like to close the session? A final comment, a vision? Your top 10 advice, finally.
Top 10 advice, yeah, give the whole presentation.
I know.
I hear a common theme, and I've also heard it voiced by the speakers, is take your mindset away from this black box that the regulators are our enemy. They're out there. If you're in nanomedicine for oncology products, those regulators have brothers and sisters and friends and relatives and neighbors that also suffer from cancer. They want these products in the market. So I said collegial, and it truly is. And resources are available for you. You don't have to be an expert in regulatory sciences. You can outsource most of that, because filing a ECTD is a lot of paperwork. Generating the CMC data is a lot of work, but that's where you come in. So continue. You're doing a great job. So let's get more products into the market.
Fantastic. I have a final advice. Come to the Metrino workshop tomorrow at 2:30 p.m. where you will learn the money we will spend to develop SOPs, reference materials. Fanny is the coordinator of this great project. We are happy to participate. It was the first attempt of a regulatory session on an ETPN event. It was fantastic to hear you. I hope you've enjoyed. Please, a round of applause for our speakers. And special thanks to Arpan and Laurent that has been participating at distance for 2 days. I mean, taking time of your busy agendas. We truly appreciate. Thanks for the effort and the involvement, guys.
Thank you, everyone. It was a pleasure.
Okay, now, short break, but you have coffee already. You don't have to wait for the coffee.